Hello, students. Today we have to learn antibody engineering. So, for antibody engineering, the first thing is that why we have to engineer antibodies. So, antibody engineering is important because we first immunize mouse and we produce antibodies in the mouse. So, the antibodies that are produced in the mouse, they will not perform better or they will not perform optimally in human bodies because mouse belong to the rodents, although it is mammal, but this one, the FC part of the antibody or this protein which belong to the mouse, it will be different and its amino acid sequence will be different. So when we inject in the human body, it will not work optimally. So in order to make it better and to work optimally, we have to engineer it. We have to humanize these antibodies. We have to produce humanized antibody, or we have to make this chimeric human body antibody. So to produce this chimeric antibody, so here you can see in this case, you can see that this mouse antibody, this blue part that is variable region, it belongs to the mouse, and this other part, it belongs to the human beings. So this one part is from human beings. So this antibody will be termed as humanized antibody because in it we see that this constant region of light and heavy chain, it belongs to the humans. Now the question is that how we can make it. And of course, all the changes will be at the DNA level, at the gene level. So for this purpose, we need antibodies which in which one part belong to the mouse and other part belong to the humans. So for this purpose, we have to make a plasmid. In the plasmid, we have to take, so in the plasmid, we have to take this, a plasmid in which we have to take this constant region. And of course, it should not be the one plasmid. We have two proteins, that is the light chain and heavy chain. For light chain, we will have this constant region already cloned with the plasmid, and then we will clone this part of the mouse variable region. So then we will be able to get this. So in the heavy chain, there will be this variable region of the heavy chain from the mouse, and for the light chain, we have to make another construct. So in it we see that so here we see that this part belong to the mouse and this part belong to the human beings, and it is making the FC part. And we know FC part is the part which is performing the function, effector functions on antibodies. And in the light chain, we see that this variable region belongs to the mouse and this other region belongs to the human beings. And it's just protein, but the changes will be made at the DNA level. So we have to make a construct, a plasmid in which part of this will be having this sequence, DNA sequence for the constant region of the heavy chain. And then we have to ligate this part of the sequence from the heavy chain of the that belong to the mouse, and then we will ligate them, and then we will transform them, and then we will produce heavy chain. And in the same way, we will produce the light chain, another clone. And then after production of these heavy and light chain, we will overexpress them, we will purify them, and with the help of ion exchange chromatography and gel filtration chromatography. And after purification, we will conjugate these two heavy and light chains, and we'll get this antibody, complete antibody structure. And this antibody will be termed as humanized antibody because now it have variable region from the mouse and have this constant region and especially FC part from the human beings, and now it will work optimally in the human beings. Because the main purpose of making these monoclonal antibodies is that we have to use them in the diagnostics and therapeutics. So for therapeutic purpose, it should have this humanized. So either we can do this strategy. Other strategy to make humanized antibodies will be that we may create a strain of the mouse in which we exchange or remove these genes which are producing this constant region, and we'll replace it with the human genes which are producing the constant region. And when that mouse will produce the antibodies, those antibodies will be already humanized. So anyway, we can make a strain and we can make this antibodies in the mouse as well. So it depends that if we have that strain or not. If we have it, then it's okay. If not, then we have to opt for some other methods. So anyway, we see that so, but here we see another strategy, that is grafted CDRs. So why we have to make this to make this grafted CDRs antibodies? Because we have this monoclonal antibody which is being produced in the mouse. In the first strategy, we have just removed this constant regions of the light and heavy chain with the help of cloning and overexpression methods with the help of recombinant DNA technology. So in this strategy, we see that this part belongs to the mouse. And when it belongs to the mouse, it will be having some immunogenicity. and it will produce some immune response because this part of the protein is foreign. It belongs to the mouse, and when we inject in the human being, then we will get some immune response. So in this case, in the case of grafted CDRs, we know that these CDRs are complementarity determining regions. Complementarity determining regions. So these complementarity determining regions are the regions which are the small segments of the variable region which show binding with the epitope. So these regions have to be retained. It should be from the mouse, and the rest of the antibody will be from the human beings. And then there will be no problems of immunogenicity. Of course, we cannot say no. It should show some, but since less amount of these sequences will be different and they belong to the mouse, so they will produce a weak immune response, but it will not be so much problem. Anyway, we can produce grafted CDRs in which we will change the small segments which belong to the mouse and which are important for binding with the epitope, and rest of the antibody will be the human. Almost it will be the complete humanized antibodies. Another strategy we see that we can produce these. Another strategy we see that we can produce this one antibody, chimeric immunotoxin. So what are toxins? So immunotoxins. So there are some toxins, for example, that belong to the Shigella or ricin or diphtheria, and these toxins will interfere with the process of protein synthesis and will kill the target cell. So immunotoxins, we can use them for the therapeutic purposes, for the treatment of tumors, for the treatment of cancers. So here you can see that this immunotoxins, this blue part belongs to the mouse in both heavy and light chains, and this part is the part which is toxin. So either we can make this or we can conjugate this toxin molecules here. And when we conjugate it here, then they will bind with the target cell, with the target tumor cell, and then their phagocytosis will take place, and then toxin will be released, and this toxin will interfere with the protein synthesis because it will stop production of elongation factor 2 and will stop the protein synthesis process. And when there will be no protein synthesis, it will result in the death of the target cell. So we can target tumor cells with the help of immunotoxins, and we can kill those target cells with the help of this strategy. So you can see that most of the part of this antibody belongs to the mouse and a small part belongs to the toxin, or we may conjugate this purified toxin with a part of this FC part, and then we can use it as immunotoxin. So again, we see that the starting point is, again, we can see that our starting point is this one, antibody which is produced in the mouse. And then we can use to make heteroconjugate. In heteroconjugate, we see that there are two arms. That is anti-T cell receptor and anti-tumor antibody. So we can use it for the treatment of tumors to kill tumor cells. So one arm of the antibody is anti-tumor and other arm is anti-T cell receptors. That means that it will activate, it will bind with the anti-T tumor T cell and will help us to conjugate these two parts, that is tumor cell and T cell, and then T cell activation will take place and it will help to destroy tumors or kill tumors. So we can use it for therapeutic strategies. So anyway, in the antibody engineering, we have discussed that initially the antibodies will be produced in the mouse, these monoclonal antibodies, and then we can modify and we can increase their efficiency by making them humanized, or we can go for grafted CDRs, or we can go for chimeric immunotoxins and heteroconjugates. So, and we can use these antibodies as therapeutic agents. We can use them for the therapy. We can use them for diagnostic purpose, and it's a very good tool. And we can make better antibodies with the help of antibody engineering. Thank you. See you later.